Hello everyone, today we're going to be going over, well, this short video I guess, we're going to go over basically looking at some of our tests. This is going to be a first in a series of 3 tests for Staphylococcus, Streptococcus, and Enterococcus. The base video you should already have watched on YouTube. This is an extension of that, looking at our pictures and how we would go about identifying exactly what it is we're looking for on these plates. Also, it should be noted that we do have a working plate reader, so I did include a plate reader, which will really help with the hemolysis test and seeing exactly what type of hemolysis is going on. So with that in mind, we're going to start with the hemolysis test. I expect this to be a fairly short video, maybe 3 to 5 minutes at max, as I go through here and show you exactly what we're looking for. So this is the results that you will have. Here's the drop page. Coding for our individual plates can be found on the lab guides and worksheets. And I do want to state the plate reader was used for the hemolysis test, except for one. There's one that it wasn't, and that's because of Enterococcus, and it's due to the nature of Enterococcus. I will explain what I mean when we get over to the Enterococcus. So if we take a look, here is the one that you can really see our coding on. And as usual, it's mirrored. So this is SS, which is Staphylococcus saprophyticus. This here is Staphylococcus aureus, and this here is Staphylococcus epidermidis. And let me pull up my annotation, because for this you can really see exactly what it is we're looking for in terms of halos. This area here, that's weird. This halo here, this area of clearing, that's what we're looking for in most of these. This is indicative of alpha hemolysis. No, beta, full hemolysis. Sometimes I get them confused. This is beta hemolysis or complete hemolysis. And we know it's complete because you have the bacteria here, which is this area here. And then around the outside of it, you have a zone of complete clearing. Unlike what you have for over here for Staphylococcus aureus and Staphylococcus epidermidis. Keep in mind that this is after 24 hours of incubation, and for some of these, they don't exactly perfectly work. We can see that here with Staphylococcus aureus, we have on this side mostly, you have 3 zones. You have your bacteria right here. That's what I find interesting. You have your second zone right here, which is actually alpha, or green hemolysis. You can see the green easier on the other side. It's this area right here, that sort of dull green. That's what we're looking for when we're looking for alpha hemolysis. And then you have this very thin area around the edge that is fully cleared, which is complete or beta hemolysis. How do we go about interpreting this? I personally would interpret this as alpha, since alpha for Staphylococcus aureus seems to be the biggest. And if we look here, the alpha zone of clearing is this entire area right here. That's the largest zone of clearing. So I would say for Staphylococcus aureus, looking at this, it would be green hemolysis or alpha hemolysis. Same thing with Staphylococcus epidermidis, even though we do have a zone of growth right here. We have that green halo. And then we have a very thin zone of alpha hemolysis on the edge. I would, for both of these, consider this to be right on the here. The green area is alpha, and the outer edge is beta. This is easier to see in other species. Definitely, this is its own thing. You can see the large clearing zone. So let's take a look then at something where the green hemolysis is easier to see. Now, when we're looking at this is our Streptococcus, and you can see here, this is the Enterococcus faecalis. When the light's coming through, it looks like there's not any growth. There is growth. It's gamma hemolytic, so it's going to be really difficult to see. And what we're seeing here on the plate reader, you can see here this large band of green, which is exactly what you're looking for on the Streptococcus feces. This large area of a dull green on the outside of a colony growth, which is right in the center. Now this is without light, and as you can see, it's much more difficult to see, though you are able to see here, this zone here of beta hemolysis, of full hemolysis, but you really don't see any color here. This is why we have the plate reader, because the plate reader allows that green to pop, and you're really able to see that green hemolysis as part of the alpha partial hemolysis. And you can easily see it on here, the beta hemolysis right on there. And this is the Streptococcus Enterococcus plate without light, mostly to show you this right here. This is gamma hemolysis. There's growth, but it's not actually breaking down the red blood cells. And that is our hemolysis video. There will be 2 other videos, one on bile escalin and what we're actually looking for the bile escalin test, and then another on the mannitol salt and what we're looking for the mannitol salt plate.